Hi, I am Mrs. Sloan, and this is video number 2 for our chapter 18, which is discussing the origin and history of life. So video 1, I already discussed the evolution, the chemical evolution and biological evolution to form the first cell. So here in video number 2, we're going to talk about the geologic time scale, the evolution of life, and we'll look at geologic factors that influence evolution. So let me make myself a little bit smaller here. So as always, the descriptors, in the descriptor of this video, I have a link to the group shared notes that my students use. So we are starting in 18.2, the history of life. And fossils tell a story, and fossils document the history of life. So the majority of fossils are found in sedimentary rock. You're going to need those for your notes. Majority of fossils found in sedimentary rock. And the assumption that is the layers on the bottom are the oldest layers and layers on the top are the newest layers. Of course, when we talk about plate tectonics, we can talk about subduction zones. So you have to be careful when you're analyzing those rock layers. And in a nutshell, if we take all of the Earth's time for perspective and put it on a 24-hour clock, I want you to think about what you were doing last night at midnight. Hopefully it was sleeping. And so we'll say that that is about 4.6 billion years ago when the Earth formed. So at 12 midnight, the Earth forms. You could write 4.6 billion years ago on your notes if you wanted to. And then you can see oldest known rocks. And then we're in what's called the Precambrian time. And about 5 a.m. This morning, remember Earth started at midnight last night, 5 a.m. this morning is when you would have your first cell. You would have a prokaryotic heterotroph. So that's about 3.5 billion years ago. And then you can see first photosynthetic organisms. It's 8, 9, 10. Here we are at 12 noon. And it's not till 1 or 2 p.m. you even have free oxygen in the atmosphere. It's not until 4 p.m. that you have a eukaryote as opposed to a prokaryote. So 4 p.m. you have eukaryotes about 2.1 billion years ago. And it's 6, we're still in the Precambrian time. 7, 8 p.m. You have multicellularity. So that would be about 1.2 billion years ago. So just for perspective, we started at midnight. We went all night into the morning, afternoon, it's 8 p.m. until we even have multicellularity. About 10 p.m. is when you have organisms that are up on land. So invasion of land, that would be about 500 million years ago. And then 11:59 and 30 seconds. So our 24-hour clock, 30 seconds before that is when you have humans. And like early humans, maybe 2.5 million years ago. And, there's some debate on all of that, but we'll say that's about 2.5 million years ago. So that is for perspective. Besides sedimentary rock, you can find fossils in tar, ice, bogs, and we've talked a little bit about this before in amber as well. And I think you have all the notes for that. So now we need to talk about how old are these fossils. So there's 2 mechanisms for that, and you can use a combination of both. Relative dating is looking at those sedimentary rock layers and assuming the older layers are on the bottom and newer layers on top. And then absolute dating is looking at the actual age of that rock. And so you use radioactive dating because the rate of decay is fixed. And we'll talk a little bit more about that. I want to point out here, I showed you a fossil in class of a trilobite. That's considered an index fossil because you only find it during the Paleozoic era because then they became extinct. So that can help you because any rock layer above that would then be newer. Right? And using a combination of both these types of dating really helps you tell the story. All right. So for dating, relative is older layers at the bottom. And then I mentioned index fossils identify deposits made at the same time worldwide. They can be used. All right, so now let's talk about absolute dating. This relies on radiometric techniques and the speed of isotope breakdown. So when you look at an isotope and it's going to break down to a more stable form, they know how long it takes to do that. And for any isotope, when you talk about its half-life, half of it is broken down, right, into that more stable form. Because that rate of decay is known. And so what you can do, here you go. So you can see you have this newly formed mineral and you know how fast and how long it takes for it to break down to the more stable form. So you use a ratio. Let me move myself here. So if you look at a new rock form today, okay, there will be carbon-12 is the most common isotope of carbon that you would find, but there's also some carbon-14, and it would be a very small percentage relative to carbon-12. But as this rock ages and gets older, that carbon-14 is going to break down into nitrogen 14. It's going to break down into a more stable form. So let's say this is a brand new one, and this is my amount of carbon-14. As this rock ages, then you're going to get less and less of that carbon-14, and we know how fast it takes. So for instance, if you know the rate of decay for a half-life is 5,730 years, right? So if you're only seeing an eighth as much, right, an eighth of the ratio present as we have today, then we know we've had one half-life, right, a half, then another 5,730 years if it is a fourth, and then another 5,700 years, right, if it is an eighth. And so you would say that would be 3 half-lives. And I think I broke it down for you here. Did I? Yes, I did. Okay. Let me move myself. So you would just take 5,700, look, I have 80 here, times 3, and you have 17,000 years old approximately, because it was 3 half-lives. So on your notes, I think I gave you everything for that. All right. So obviously you need to, when something's older than this, right, you're not gonna be able to measure that carbon any longer. So you're gonna have to use a different isotope in order. And here you can see how the half-lives, obviously you don't use something like polonium. Look how fast the half-life is, like 3 minutes. You have to use the right isotope depending on how old that substance is. Okay. So now that we know how to do that dating, we're going to look at the tree of life here, the overall. So you can see your first cell, remember, is about 3.5 billion years ago. You have bacteria and archaea. We've already talked about this. So let's just follow the bacteria out a little bit. So eventually you would have aerobic bacteria, right? So these are ones that are doing cellular respiration, using oxygen in order to generate ATP. You've got photosynthetic bacteria that now you have evolved the splitting of water, right? And so you have your 2 photosystems for your photophosphorylation. And then you could have some other photosynthetic bacteria as well. And then those would be our current bacteria. Let's go up here to the split. Archaea, remember, archaea is more similar to eukarya than bacteria. And you can see here you've got your thermophiles, your halophiles, remember those would be in salt, methane producing methanogens, but you have a split off here of eukarya. Remember, to be eukaryotic, we have the endosymbiotic hypothesis would have to occur. So you have pre-mitochondria being aerobic bacteria, pre-chloroplast for plants being photosynthetic bacteria. And we've talked before, and we'll visit this again, the endosymbiotic hypothesis, and then the formation of plants and other protists. These heterotrophic ones that don't do photosynthesis, right, would then evolve into animals, fungi, and protists. Your Paleozoic era, when we transition from Precambrian to Paleozoic, and most all time here on Earth is Precambrian, right? Because we're talking the Earth itself being about 4.6 billion years old. So that would be like 4,600 million years, right? And here the Paleozoic era would start right about here, about 543 million years ago. The first period in the Paleozoic era is called the Cambrian period. So that's why they call all time before that is Precambrian. So that's an overview. If you take a look at your notes for a minute, you can see we have in our notes the Precambrian time, and then we have the Paleozoic era, which is what I just referenced. Then the Mesozoic era is in the middle, and the era you're in right now is the Cenozoic era. So we want to have a better understanding of each of those time periods. So here in the Precambrian, just remind yourself, formation of Earth, oldest known fossils, oxygen accumulates, we have eukaryotes. Now we have protists, right? Those are primarily single-celled eukaryotes. And then Ediacaran are like small multicellular invertebrates, like flatworms. And then here we have the Cambrian animals of the Paleozoic era. So on our notes, for Precambrian time, I want you to have 87% of the total geologic time scale. 87%. Okay, that's most time of this Earth. The first cells about 3.5 billion years ago. So stromatolites. Before I talk about stromatolites, you know how like when a tree is cut down and you can see the rings in the tree and you can go back in time? You can do that with bacteria with these stromatolites. They exist today. These are obviously pictures today. And they will cut into them and it's like going back in time. What bacteria do, cyanobacteria, which probably contributed to the oxygen revolution more than likely, they would keep moving outwards. They'd have a growth of bacteria and the mucus and the slime, the sand would get caught in it. It would start to harden and calcify. And so the living bacteria would keep being on the edges, on the edges, on the edges, on the edges. And it's like the history of cyanobacteria back in time. And so they look at those. So I have stromatolites. So bacteria, sand, boulders still exist today. And the outer edges are still generating oxygen. But that's kind of going back in time when you cut into those. The oxygen revolution, as I mentioned in video 1, all Earth didn't have, for the bulk of time, did not have any free oxygen of any great quantity. It was absorbed into rocks in the ocean. So when the oxygen revolution occurred, when water was split to release that oxygen, huge mass extinctions because it was considered a toxin, it was a poison. But with every extinction, we're going to talk about it, it makes available all these habitats for new life to diversify. And so whenever there's a mass extinction, you see massive, rapid new life forms evolving from that. So on your oxygen revolution about 2 billion years ago, when that occurred though, then that's when the ozone layer formed, and that ozone layer was very protective of life and blocking out some of that UV radiation so that life could crawl out of the waters and get onto land. So next, and I think I gave you everything for that. Ozone shield is what you want, a generating protective ozone shield. Okay, a review of the endosymbiotic hypothesis. We have already learned this, so hopefully this is just a good review. So the idea here is how you would go from a prokaryotic cell to a eukaryotic cell. Put a pin in mitochondria and chloroplast. I'll come back to that in just a minute. But remember, you have membrane enclosed organelles, specifically your DNA is enclosed. So remember we learned about with cell membranes, endo and exocytosis, so that infolding of a membrane could go and be for those cells that had that infolding of that membrane around their genetic material, their stored DNA, then they would probably be more likely to survive and reproduce with their DNA protected like that, yes. And then those membranes could have formed your ER, your rough and smooth ER. A symbiotic relationship of one prokaryote living inside of another prokaryote, that would be the origins of both mitochondria and chloroplasts. What's our evidence for that? Both mitochondria and chloroplasts are the right size. They have their own DNA. It is circular, just like prokaryotic DNA. There are no histones, just like bacteria. They are self-replicating by binary fission. They can do their own protein synthesis, and they have a double membrane, which would lead evidence that they had been engulfed. So on your notes, eukaryotic cell 2 billion years ago, endosymbiotic hypothesis, I gave you all of that. Then little letter A, they are the right size. They have their own DNA, not packaged on histones. It's circular, self-replicating, divide by binary fission. They have two membranes, an outer membrane like eukaryotes and an inner membrane like prokaryotes. All right. Next, multicellularity about 1.2 billion years ago. I'm trying to put myself over here. So you can see here, this kind of would remind you, remember when we talked about development and we talked about the blastula and then the infolding, right? So you can kind of see that mirrored here, those homologies in order to form a multicellular organism, that infolding right here. Initially 2 layers, no mouth, and then that gave rise to other invertebrates. So for your notes, I think I gave you everything for that. So we're good. Moving on. So now let's look to, so everything we just did was Precambrian. So now we're going to move into, and that was 87% of all life. So now we're going to look at the Paleozoic, Mesozoic, and Cenozoic. And what I found is if you can get the broad outlines of each of these eras and think of them as of course I can't think of this, bookends, right? So if you know where it started and where it ended, then you can kind of look at what life would have evolved through that. And then you just know the bookends and then you'll be good and know those time periods. So if we look at Paleozoic era, that's about 542 million years ago to 251 million years. So it goes from 251 all the way back to 542 million years ago. So what did you see there? Major developments. You went from algae to, and obviously I'm jumping over some other milestones, to things like ferns, all the way up to gymnosperms. And these are like your Christmas trees or your Hanukkah bushes. So major life changes there for plants. And then animals, you're going from invertebrates to arthropods to vertebrates and fish, and from fish, amphibians to early reptiles. So big changes there. Once you go into the Mesozoic era, that started about 251 million years ago. You're taking gymnosperms all the way up to angiosperms. Angiosperms are any plants with flowers. And then animal-wise, you're going from reptiles and birds all the way to your early mammals. And then the Cenozoic era is the center of your world right now. You have angiosperms up to small herb or herbaceous plants, and early mammals all the way up to humans. So the reason why I wrote the numbers the way I wrote them here is right now is the Cenozoic era, and that goes back about 66 million years ago. And then the Mesozoic era started about 66 million years ago, and that goes back to about 251 million years ago. And then your Paleozoic era is 251 million years ago is when it ended, and it goes back all the way to 542 million years ago. And everything before that is your Precambrian, right? Okay, so that's your big picture. So on your Paleozoic era, we're going to start here and work our way through these three. When you look here, you can see, let's follow this line up as plant life, and this one is animal life. So you can see like first plants, marine algae are flourishing, mass extinction. Now you're starting to get seedless vascular plants, first seed plants appear, mass extinction. Then you're starting to get forests and giant gymnosperms as a result of that. Animal life, you're going from invertebrates, your first chordates, jawless fish, mass extinctions, jawed fish, and then first amphibians, mass extinction, and then amphibians, and then reptiles. So that's your big picture. And we're going to jump right in here to Cambrian animals, because what I want you to know from that, we're in the Paleozoic era. The first period is the Cambrian period. All life, this is key, All life, that's what goes in your notes, traces its ancestry to this Cambrian explosion of life, Cambrian explosions. Animals had protective exoskeletons, fossilized well, and here's the beginning, you can see, of your trilobite. When here I have another one, trilobite. So there are over 20,000 species of trilobites, and you want to know that's your key index fossil. At the end of the Paleozoic era, that mass extinction is called the Great Dying, where anywhere between 83-95% of all life over. And then you have a whole new great diversification of life coming after the end of the Paleozoic era. So that, I think we got everything there. So next, what I want to talk to you about is the molecular clock. You don't measure time in this clock in seconds, but on the rate of mutations, which is considered a very steady rate of mutations, not affected by natural selection. So if you know in this question I'm posing here that typically you would get 5 mutations every million years, then how long ago did 2 organisms diverge if there are 20 mutations that separate them, right? Well, so you have 20 mutations divided by 5 mutations every year, then yes, you're at about 4 million years ago is when you diverged from each other. So this is what's called a molecular clock. So on your notes, I think you have molecular clock based on the principle that mutations in certain parts of the genome occur at a fixed rate and are not tied to natural selection. So the number of base pair differences indicates how long 2 species have evolved separately. So you can use that now in living organisms, right? So we're not looking at, I mean, you can still use that with relative and absolute dating, right? With rocks, but this is another way you can measure that. Okay, so in the Paleozoic era, 542, which I think of as dinner time, and 248 is when I think of school's out. That helps remember that. If you think about your afternoon, right? So you go all the way from algae to gymnosperms in the plant world and invertebrates all the way to your reptiles, your early reptiles. So invasion of land over a long period, algae to ferns to gymnosperms. That's what goes in your notes. And then invertebrates, arthropods, vertebrates, fish, amphibians to early reptiles. So those are your 2 bookends. All right. Next, insects. This is huge because having insects, huge diversification of insects during the Paleozoic era. And this is important because this is setting us up for the evolution of angiosperms later, right? If you have a combustion engine, you are using the plants that they're at towards the end of the Paleozoic era, and you're using that as coal. And yeah, and are we going to run out? Yeah, eventually we are. So we better think of some other options, right? Several 3 mass extinctions that we looked at. But at the end of the Paleozoic era, this is called the Great Dying. So let's just round it to about 9, because there's a window of how many died, but let's just say 90% possibly, maybe 83, maybe more than 90%. Large-scale volcanic activity could have led to it, increased carbon dioxide, significant global warming, and wiped out almost all life. All right, so on your notes, I have that tied in at the end where you can see, if you look to the end of your notes in 18.3 with mass extinctions, number 2, under mass extinctions, under 18.3, there were 3 in the Paleozoic era ending with the Great Dying. So somewhere 83-90% of all life, maybe a little more than that. So that is located there in your notes, okay? All right, Mesozoic era is in the middle. As far as what do you have during the Mesozoic era, so let's take a look at this right here. So you have forests, conifers, and you have a mass extinction, and what comes about? Flowering plants. And flowering plants are called angiosperms. Then you look here, first mammals are starting, dinosaurs. Dinosaurs are flourishing when? during the Jurassic period, like the movie. And then you have even more mammals after a mass extinction here. Then we're going to just take off here in the Cenozoic era. So as far as your notes go, you have 251 to about 65 million years ago, plants, gymnosperms, and the beginnings of angiosperms, and you have reptiles to early mammals, So this is the Triassic period. Let me go back one right here. So it goes Triassic, Jurassic, and Cretaceous. So here's your Triassic types of plants you would find then that still exist today. Here's your Jurassic period from one of the movies. If you like dinosaurs, this is when they're doing that. Okay, and then you have your early mammals. All right, so another mass extinction at the end, making possible for the adaptive radiation of mammals and an explosion of mammals here in the Cenozoic era. All right, so here we go. You can see some of the organisms that might exist then. And on your notes, let's see. Here we have some early primates and eventually to humans. So on your notes, plants, you have angiosperms to herbs, and then animals go mammals to humans. And this was a time of megafauna, giant ground sloths, wolves, bison, mammoths. And yeah, here's another character from a movie. So here is megafauna. All right, and that's the end of it for Cenozoic era. And now the last part of this chapter, geologic factors that influence evolution. Most of this is review for you, so this will go pretty quick. You can see here some seafloor here, hydrothermal vents, going back to video number 1, and plate tectonics. So we're going to talk about continental drift. And continental drift explains why several continents mirror each other like puzzle pieces and have shared geologic structures, because they were once joined, have shared geologic structures. It explains the unique distribution patterns of several fossils. Like here you can see this distribution of a fern, and you can see that in the fossil record as well. Explains the unique mammalian diversity because 65 million years ago, which was the beginning of the Cenozoic era and the explosion of mammals, all the continents were separate, and that's why you have that unique mammals on each of those continents. So explains unique mammalian diversity on different continents. So why the drift? Why are these continents? And this goes back to what you learned last year and even this year on our plate tectonics. We want to remember about subduction zones, and you can see that here in this picture. Here is a subduction zone. Forward edge of one plate sinks below another, and that's where you see those volcanoes. Let's see, let's see, what else did I wanted to get there? Oh, and I talked to you about when you have a convergence of two continents, then you get things like the Himalayas. And a transform boundary is when two continents are sliding past one another. Here in California, we know that as the San Andreas Fault, which gives us our earthquake, or down in the valley, you can see the flowing of the valley floor. And so we have two plates that are trying to slide past one another. All right, now moving on. So to the end. Mass extinctions, I mentioned that a little bit earlier. I talked about the Great Dying. Here you can see the different mass extinctions, 5. 3 here in the Paleozoic era, maybe due to continental drift, maybe meteorites, CO2 levels causing these extinctions. On your notes, I think I gave you everything except here gives you another little summary. Trying to move myself out of the way here. Sorry about that. There we go. Showing you the time and what likely caused those extinctions. What I want you to remember from these is that these huge disasters, what came out of it is this radiation of new species from that. So on 5 mass extinctions, 3 in the Paleozoic era, ending with the Great Dying, 83, 90%. Here you can see they say 95%. We'll just average it to 90 of all species. There were 2 in the Mesozoic era, ending with 50% of all species becoming extinct, dinosaurs being one of them. But when these extinctions occur, new habitats open up for other species. New habitats open up for other species. For example, the extinction of dinosaurs was followed by the rapid evolution of mammals, and we call that adaptive radiation. Okay, the next thing I have in your notes, I'm just pulling in here. Let me try to move myself. Just a couple words, terms I wanted to review with you. Do you remember when we talked about evo-devo, evolutionary biology, and development? Remember, and those switches? We have heard of adaptations, but exaptations, boy, that was hard. Structures gradually playing a new role, like feathers might initially have been for regulation of body temperature, but then got co-opted to be used as wings. Another term, heterochrony, changing the rate of developmental events. Remember about those switches, back to that again, that phalanges would be longer on bats for wings. And then we talked about homeotic genes, master regulatory genes, specifically Hox genes, a class of homeotic genes. Small changes, because these are transcription factors, could leave snakes without legs, and etc. So they've already messed with that a little bit. I just wanted to kind of bring those terms in from earlier discussions that we have had. And I think that's it. All right. And if you're one of my students, I will see you in class.